RFLP, restriction fragment length polymorphism. It is another technique that can be used to study the polymorphic variation within the DNA sequence. Now, polymorphism term, it can be defined in different ways and it can vary from subject to subject. Now, as far as the course of genetics or molecular biology is concerned, it is mainly dealing with the variations in DNA sequences. That the variation may be in single base pair in a particular sequence while comparison, or it may be due to indels. Indels mean a particular DNA sequence it has deleted or added. So it is more related to variations in a DNA sequence, and it is called as polymorphism or polymorphic DNA. And such differences within a genomic DNA we can study by using RFLP technique, that is restriction fragment length polymorphism. Why the name restriction it is applied? Now we are going to discuss. For example, अगर हम human genome को compare करें, तो more or less as far as sequence is concerned, तो human genome जो है वो 99.9% जो है वो similar होता है, और overall जो different sequence में report किया गया है वो 0.1% तक हमारे knowledge में है. कुछ literature में 0.5 तक भी है, but mainly it is around 0.1% variation in DNA sequence. Overall, 99.9% it is the same. But still this difference, that is 0.1%, it can be used or exploited in order to find the variations among different individuals of humans. Not only humans, other living organisms. अब इन वेरिएशन का क्या फ़ायदा होता है कि जो 0.1% वेरिएशन है ह्यूमन जीनोम में, how it is beneficial, how it can be used, and why these variations it can be used to study different matters related to human daily life. Suppose 0.1% variation is there, and it may be that 1000 base pair may have one nucleotide change. So when one nucleotide changes there among 1000 nucleotides from individual to individual, then the target sites for different restriction enzyme it can also be changed. So these slight changes like 0.1%, it can influence the restriction pattern of the genomic DNA. मिसाल के तौर पर अगर हमने ह्यूमन जीनोम को एक्सट्रैक्ट किया है, एक्सट्रैक्शन के बाद उसको अगर हम डाइजेस्ट करें किसी ख़ास रिस्ट्रिक्शन एंजाइम से, लाइक EcoR1 से, BAMH1 से, तो उस डाइजेशन के नतीजे में क्या होगा कि छोटे छोटे फ्रैगमेंट्स बन जाएंगे DNA के, और उन फ्रैगमेंट्स को लेटर ऑन हम सेपरेट कर सकते हैं ऑन द जेल इलेक्ट्रोफोरेसिस. So depending upon the length of the DNA fragments, we will get a particular binding patterns. So on the basis of length, DNA fragments, they will be resolved or separated on the gel. Now, we want to know that whether the human individuals, they are showing different restriction pattern. We can also use this strategy that the two individual fragment restriction pattern it can be studied, and due to 0.1% difference it will be slightly variable. Now this restriction pattern it has been used extensively in the study of forensic sciences, where a particular individual it can be linked with certain crimes or legal matters. Initially fingerprinting was used, but later on due to the advancement in the techniques of molecular biology, now DNA fingerprinting technique was developed, and this was based on DNA profiling or restriction fragment length polymorphism or DNA pattern that is due to the variation in the DNA fragments, because the restriction sites, they may be located at different intervals that will produce a different banding pattern. Now, how this technique it can be used in forensic sciences, although we will discuss this application slightly later on, but here I can introduce the brief outline. Now, suppose we have some suspect that may be linked with certain crimes or legal matters, and we have extracted the genomic DNA from the suspect, and after extraction it can be cleaved with certain restriction enzymes in second step, so that after restriction we will get a number of DNA fragments, and these DNA fragments it can be separated by agarose gel electrophoresis. And in the next step, when the DNA fragments are produced, they can be resolved on the gel, and it can be compared with different markers. For example, in the first two lanes, DNA markers are there on the right and left, and we have DNA from the suspect that can be compared with the evidence source. For example, we have collected blood sample tissue from the crime, and this DNA it can also be extracted and it can be resolved along with suspected victim. Like suspect and victim DNA, they are run together, and different suspects DNA can be loaded on the gel, and after running this pattern it can be resolved on the gel, and banding pattern can be compared to know that whether criminal they are associated with this particular crime or not. And after the separation on the gel, these DNA fragments can also be transferred for southern blot analysis on the nylon membrane paper. And in the next step, when it is transferred to the nylon membrane, then fragments they can be detected by using certain radio labeled probes. So probe hybridization is there, the technique you are well aware, and then after hybridization it can be exposed to X-ray film so that a particular DNA bending pattern it is available. So by using RFLP technique, we can resolve the DNA fragment that are called as restriction fragments, and it has application in the field of forensic sciences, where we can know that whether a particular suspect it has committed the crime or not.